In this lesson, we're going to jump through some of the steps and the purpose behind the use of recombinant DNA technology. So before we can really get into recombinant DNA technology, we need to unpack this idea of a synthetic hormone. So generally speaking, these are chemicals or hormones which are identical to the natural hormones, but instead of being produced by the body, they've been produced outside the body, aka they've been produced in a lab. Now, the way that we go about producing these synthetic hormones is through a process of recombinant DNA technology. So hormones produced through the use of recombinant DNA tech generally have minimal side effects, which makes them highly effective. And the reason that they have minimal side effects is they are produced using the real human gene. So what is recombinant DNA technology? Well, it's DNA that is formed artificially, and this is our key idea here. It's not natural, it's an artificial process that we induce, and we do it by combining genes from different organisms. Now, why do we do this? Well, as we've just mentioned, we can make synthetic hormones, okay? And we can get them to make copies of a particular gene in some organisms or the products that it codes for. So, for instance, this is being used to treat cystic fibrosis, it's being used to help us create vaccines, it's being used to help us create, and I'll note here, synthetic hormones. Okay. So, this here is the overall process, and we're going to dive down into 10 key steps that you need to know for your exam or your test. But the main step here is we isolate a particular gene that we want from the human cell. Okay, why a human cell? Well, this is going to give us the most minimal side effects possible. So in this case, the gene that we've isolated is insulin. Okay, so we're looking at the control of blood glucose. Now, just as we've taken a gene from a human cell, and I'll highlight this in yellow, and eventually over here as well, we've also taken a plasmid from a bacteria. Now, a plasmid is a circular form of DNA that's found within bacterial cells. And what we've done to both the insulin gene and to the plasmid is we have cut it, okay, or cut them with the same restriction enzyme. Now, I'll unpack what a restriction enzyme is in a bit more detail in a second, but this restriction enzyme, you can think of like a pair of scissors, and it produces a sticky end. Now, what I mean by this, as we've been through previously, is a cut which is non-straight and produces these fragments, okay, towards the end of the DNA sequence. Now, what we can do is we can introduce the insulin gene to the plasmid, and we can seal this off with some DNA ligase. And what's going to happen here is we end up with our recombinant plasmid. So maybe I'll highlight that one here in blue. We then take the recombinant plasmid and we insert it back into that original bacteria. So you'll notice here, we've placed the plasmid back into the bacteria. And it's now known as a transgenic bacteria. Transgenic meaning multiple genes, okay, from multiple different species, aka the human cell and the bacterial cell. Now, what we then do is we culture these bacteria, okay? We allow them to replicate. And what they're going to do is, because they now contain this gene to produce insulin, when we've grown a large number of these bacteria, they're going to produce a large amount of insulin. And then our final step, the really key part for us, is that we can then extract the insulin that they've made. We're not extracting the bacteria itself. We want their product in this case. We want the insulin. So let's break this down. So when we're cutting the DNA, I've mentioned these things known as restriction enzymes, right? So in order to use a restriction enzyme, we firstly need to isolate the gene that we want, and we cut it using the restriction enzyme, okay? Now, we're always going to cut the DNA at points where there's a specific sequence of bases, and this is what we call a recognition site. It's how we know where to cut, okay? Now, some restriction enzymes are going to produce a straight cut or blunt ends. So blunt ends are like my example here, where I've got my DNA sequence and I've cut it directly through the middle. Okay, so you know what, maybe I'll switch this over to green here. This is my blunt ends. So this is a straight cut. And in pink, I'll use what we call a staggered cut in order to produce sticky ends. So we can see this cut in between our sequence here. Now, sticky ends allow us to really easily combine with other sections of DNA that have a complementary ending. Okay. So as we come down here, here's another representation. You might like to pause and just have a read through, but for now we're going to skip over. So DNA ligase. You see I've forgotten to erase my annotation here, but effectively DNA ligase is going to act like our glue. If our restriction enzyme is our scissors, our DNA ligase is our glue. And why is it our glue? Well, it's what we use to join pieces of DNA together in a process known as ligation. It's important to note that the ligation itself is with the sugar phosphate backbone, okay? It's where the phosphate molecule connects to the sugar. This is the bond that is restored using DNA ligase. And what we're going to do in this instance is we take sticky ends from two different DNA fragments and we can combine them together. So, as mentioned earlier, here's our human gene. Let's say it's for insulin again. And here's our plasmid that we took from a bacterial cell. You'll notice here that on either end of this gene, we have these sticky ends, and I have the complementary sticky ends on the surface of the plasmid as well. I can then use DNA ligase to glue these together, and we notice in the final product over here that I've just circled in orange, we have our recombinant DNA. We've introduced the gene into that plasmid. So, another name for a plasmid, okay, is a vector, okay? So, a vector is a broader term, and our plasmid is an example of a vector, okay? So, for instance, here we can see our plasmid that I've just highlighted in yellow there. The vector is the plasmid, okay? Now, when we're talking about a vector, the reason I've included it in this slide here is it is a word that can come up throughout test papers and exams, okay? And really simply, we're going to define it as a DNA molecule that is used to carry DNA into a target cell. And in this case, it'll be into a bacterial cell, okay? So, here's a different representation of some of the steps, but what I'm going to do is I'm going to go through 10 steps now. So I've got 5 on this slide, 5 on the next slide. So to start us off, our first step in recombinant DNA is isolation. We need to isolate the particular gene that we want. Secondly, we cut this gene and remove it from the cell's DNA using a restriction enzyme. Okay? We're not cutting the entire sequence of DNA, we're just cutting that particular gene. Okay? Now this restriction enzyme is only going to cut at a specific recognition site. Okay? And what it's going to produce, hopefully for us, is some sticky ends. So those ends which are overlapping. Okay? With our fourth step, we're now going to repeat this process, but we're going to isolate a bacterial plasmid. And what we then do with this bacterial plasmid is we cut it, note, with the same restriction enzyme. Okay? And this produces complementary, it produces sticky ends which are going to fit perfectly with the sticky ends produced when we've cut our gene from our target cell. Now, the human DNA gene is then going to be joined at the sticky ends to develop those hydrogen bonds between the complementary nucleotides. And what we're going to end up with at the end of this is some recombinant DNA. So, the DNA ligase is going to stick the backbone together, recombinant plasmid, where we've reintroduced our recombinant DNA, the recombinant plasmid itself is placed back into the bacteria. The bacteria then are cultured or multiply, okay? We like to use the word cultured, so please make sure that you include that in your responses. And large amounts of the bacteria produce large amounts of the desired protein or hormone, e.g. insulin, okay? What we then do is we can harvest, we can purify, and we can then provide this harvested hormone, in this case insulin, to diabetics who require this particular hormone, or for what other hormone we might use. So what I've got on the next 2 slides is just some more representations. Feel free to pause on either of these 2 representations. And then the final thing that I've included right at the end, which I'd encourage you to have a read through, is number one, an example of how we can create a vaccine for hepatitis B using this recombinant DNA technology. And number two, how we can create insulin. So the process that we largely looked at towards the start of the lesson. But altogether, this is recombinant DNA tech.